I'm Stanley, and I'm going to talk about our work here in Utrecht regarding small vessel disease and for 7-Tesla MRI. So I'll start with briefly introducing small vessel disease. I don't think it's new for many people here, regardless. It has to be present mode. Okay. There we go. This is better, I think, right? Small vessel disease. It's a disorder, pathophysiology that concerns the small arterioles, capillaries, and vessels in the brain. And typically, people with the small vessel disease, they present themselves in the clinic with memory problems, cognitive impairment, and typically what we see on clinical MRIs are these lesions in the white matter, such as these white matter hyperintensities. Let me have a pointer. So these white matter hypertensives here and these lacunar infarcts. And we know that small vessel disease is a big cause of strokes and dementia worldwide. And typically what we know is that whatever we see as white matter damage in the brain is actually more of a downstream consequence of a disease process that's actually happening in these small arterioles themselves. So ideally, we want to measure or be able to probe the arterioles or small vessel disease directly at the core of the disease, which would be these arteries. Because we believe that whatever is happening in these small vessels will eventually cause the damage and the cognitive impairment that comes with it. So we want to know what exactly is the disease process in these small vessels. And with regular clinical 3-Tesla MRI, it's not really possible to in vivo measure the small vessel functions directly at the level of these small vessels. However, with the advantage of high field MRI, of 7-Tesla MRI, we can do that. We can directly measure the state, the condition of these vessels and see how that can give us information on how they relate to visible injury in small vessel disease. So here in Utrecht, we started the Zoom at SVD study, which had as main goal to investigate which aspects of small vessel function, measurements of the state of these vessels on 7-Tesla MRI are affected in several forms of small vessel disease. In this case, a genetic variant and a more or less acquired variant, which does not have a real defined etiology, whereas the genetic variant obviously is originated from a mutation. And the study design of this study, I'll briefly walk you through it. It's an observational cohort study with these two patient populations. First, the genetic variant of the disease is called CADASIL. These are people, these were recruited from a partner center of the study in Munich. And then we had our own sporadic SVD patients who originated from our own memory cohorts. For the rest of my talk and for the purpose of this study, I'll only focus on the genetic variant of the study. So we'll only talk about the CADASIL participants. All participants underwent a baseline visit where we did medical history, performed a regular 3-Tesla clinical MRI with the purpose of measuring the disease burden, so what the white matter damage was at that point of time. We performed neuropsychological assessments and blood pressure monitoring. Then 2 weeks later, all participants were invited to Utrecht to undergo a 7-Tesla MRI. That means that even the CADASIL participants from Germany went to Utrecht. And here we performed several small vessel function measures, which I will talk about later. And then 2 years later, we repeated some of the baseline visit measurements. So we repeated the 3-Tesla MRI and the neuropsychological assessments to see if there has been any longitudinal changes in these participants. And like I said, for this talk, I'll only focus on just the genetic variant of small vessel disease. So what did we do? What did we measure on 7-Tesla MRI here in Utrecht? Jaco already alluded to this earlier this morning. We measured blood flow velocity and pulsatility on a 2D slice at the level of the perforating arteries at the base of ganglia. and they're perforating arteries at the level of the semioval center. Here in image A, you see a T1 weighted image where I've highlighted these two slices, the plannings of these slices. Now, what's fascinating here is that, especially in the slice of the center semioval, which is image B here, these arterioles are, well, probably the size of a few hundred microns. And we're able to detect these using this 7 Tesla and the phase contrast sequence. Not only can we detect these, we can also measure the blood flow velocity and the pulsatility of the blood flow velocity in those arteries. So, and we can measure the velocity of the cardiac cycle, which is denoted in image D here. So this is the blood flow velocity in the mean blood flow velocity of all the detected arteries in the center semioval. And we can do the same thing in the basal ganglia. Here, the arterioles are, well, 3 times larger, approximately a millimeter. We can do the same thing and note that the velocities here are a little bit higher than the velocities as in the center semioval. So with these cardiac velocity traces over time, we can measure the pulsatility index, which is just the difference between the normalized maximum velocity and the minimum velocity divided by the mean. And these are important markers for blood flow velocity that we measured in the ZOOMIT SPD study. And these markers primarily reflect the state of the arterioles, whereas in small vessel disease, we're also interested in other areas or aspects of the cardiovascular tree. So the secondary and complementary measure that we also performed on Zoom was looking at the whole brain CVR, bold CVR, which is obviously more venous weighted, using hypercapnic stimulus as our well, basically as our activation. So what we did here is we had a sequence of 10 minutes where we alternated 2 minutes of medical air with 2 minutes of hypercapnic gas. The gas was 6% CO2, the rest was filled with room air, and then we alternated this, and this was our regressor for the bold CVR analysis. And then we did this on the whole brain level using 70, and where we also tried to mask out the large draining veins to really look at the bold CVR of the tissue of the small vessels. And using the CVR, we can then, well, using the bold change over time, we can obviously measure the CVR, which is also what we measured on a voxel-wise basis in these participants. Then finally, the last small vessel function measure that we performed was specifically the cerebrovascular reactivity in the visual cortex due to a visual stimulus. This dartboard here, all participants were tasked to look at the dartboard. This was flickering in different frequencies and patterns. And this obviously evoked a neuronal activity in the visual cortex, which then through neurovascular coupling leads to this vascular dilatation in the visual cortex, increased CVR, which can be picked up with the bold signal. And this can then be quantified using a hemodynamic response function, as shown here, where we then look at the peak of the and the full width at half max of this fitted HRF as our outcome measures. So we already did these measurements in this cohort, and I'll briefly summarize the results of what was already done. First of all, look at the demographics of our patients. So we included 23 CADASIL patients from Germany, and we found 13 age and sex match controls. It went fairly well. The age and sex matching a little bit, the CADASIL patients were a little bit older. Looking at the vascular risk profile, I don't think it comes as a surprise that the vascular risk burden was higher in our CADASIL population compared to controls, as well as the imaging markers. So if you look at, for instance, the white matter hyperintensity volume, it's all as a percentage of the intracranial volume. And you can see that the burden in our CADASIL group was much higher when compared to our healthy controls. So what were the results that we found? So the small vessel function at baseline in CADASIL, we found that when compared to healthy controls, CADASIL patients had a lower blood flow velocity, but an increased pulsatility in the small perforating arterioles. For instance, here you see a figure of the flow velocity in the basal ganglia. What we also saw was that if you look at the visual hemodynamic response function, the bolt CVR, the CVR of the bolt in the visual cortex, you see that the HRF in the CADASIL group had a significantly lower peak when compared to healthy controls. The full width and half max was roughly the same. And if we then looked at the bolt CVR in a total white matter, the bolt CVR was not significantly higher or lower in CADASIL when compared to controls. So at baseline, we already found that different aspects of small vessel function are impaired in CADASIL, but what we wanted to know is how does small vessel function then relate to the white matter damage that we observe in the clinic. So, and that is something that we look into in this study more specifically. And for white matter injury, we took two markers. The first one is, I think, the most straightforward one. We looked into white matter hyperintensities on T2 weighted images. We just extracted the white matter hyperintensity volume. And then we did this at baseline, but also at follow-up to also track the changes of white matter hyperintensity volume over time. And then we also looked into white matter integrity using diffusion weighted imaging. And I'll briefly explain this more on the next slide. So with diffusion weighted imaging, we are looking at the white matter microstructure, which is known to be also impaired in small vessel disease. And one of the novel markers that has been established a couple of years ago, actually, is called PSND, which actually takes a look at the mean diffusivity values, which are computed from the DTI scans. And then the mean diffusivity values are then projected on a white matter tract skeleton, as shown here. And then if you then mask out regions that are either near CSF or mask out regions that are actually within lesions, you can then calculate the distribution of all your mean diffusivity voxels in this white matter tract, and then you can plot them here. And this PSND, or the peak width of the skeletonized mean diffusivity, is just the width of this distribution, which tells us something about the white matter integrity. And this is a whole brain marker, as in we can compute this distribution for all the voxels in the brain. And it is found that CADASIL patients or small vessel disease patients had a higher PSND when compared to healthy controls. So this was also something that we took as an outcome marker for white matter damage, and we did this on baseline and follow-up as well. So I briefly mentioned this already, how does small vessel function associate with white matter injury at baseline, so cross-sectionally, but more importantly, and I think more interestingly, how does small vessel function associate with progressive white matter brain injury, and meaning what happens to the brain after 2 years, and how is small vessel function maybe predictive of that? So we did this by doing univariate linear regressions between the small vessel function measures that I briefly introduced before and the two outcome measures that I just mentioned, so white matter hyperintensity volume and PSND baseline and follow-up. Looking at the baseline associations, here are the brief demographics again. What I want to highlight here in particular is that if you look at the white matter hyperintensity volume at baseline and follow-up, there has been a significant increase after 2 years. The median follow-up time was a little bit over 2 years, as well as the PSND, which also had a significant increase after 2 years. This was all done with pairwise t-tests. At baseline, we found that the full width at half max was significantly associated with white matter hyperintensity volume of the visual stimulus measured with bolt. Looking at the blood flow velocity of the arterioles and the pulsatility index with phase contrast sequence, we found that the mean blood flow velocity in the semi-oval center was significantly associated with both white matter hyperintensity volume and PSND. None of the other measures were unfortunately significantly associated. Looking at the bold CVR, if we then look at the total white matter bold CVR, so we took all the voxels of the white matter and then we averaged them over the white matter, we found no significant associations at baseline for these two outcome measures of white matter damage. Longitudinally, let's first focus on the white matter hyperintensities. Here you see a box plot where you can see the white matter hyperintensity volume change after 2 years. And if we then look at the results, unfortunately, as much as I hope to see something significant here, we could not find an association between any of our small vessel function measures and whole brain white matter hyperintensity volume change after 2 years. And for PSMD, the same story. So we did see a significant increase in PSMD after 2 years, but none of our small vessel function measures at 7 Tesla MRI were associated or significantly associated with PSND change after 2 years. Summarize, what did we learn so far in the Zoom study is that different aspects of small vessel function are affected in CADASIL. That we do know. We also know that small vessel function is associated with whole brain white matter injury, but only at baseline and not with injury progression after 2 years. However, that doesn't mean that the study is over, because there are some new aspects we can look into, future plans that we are working on right now. But first, let's focus on the limitations. So obviously, we're dealing with a limited sample size here. CADASIL is a very rare disorder, so we could not have a very large cohort of people to do these regression analyses on. We also did not formally correct for follow-up time in our analyses, as well as we did not. We only did whole brain level analyses, and I think here is where the opportunities arise, where we can maybe look into new methods and new approaches to model white matter damage progression in these people, because we can also look into voxel-wise analyses that might give us more sensitivity to model local changes to white matter. And maybe using bold CBR, which inherently we measured on a voxel-wise level, we can hopefully see if that can also tell us something about local changes and local damages that might arise in the white matter in these participants. So yeah, it's a lot of interesting stuff that we can still do, and hopefully we can also apply the same analyses on our secondary cohorts of small vessel disease patients. These results will hopefully follow soon. And that's basically the end of my presentation. I want to thank all my co-authors, the participants of the Zoom studies, and as well as all our consortium partners and the people of the Translational Neuroimaging Group here in Utrecht for their help. And if there are any questions, please let me know.